We sterilized the nutrient medium, we sterilized the explant, yes or no, right? Sir, sterilization. What it is? Sterilization. Sterilization of a nutrient medium. Sterilization of what, sir? Nutrient medium. Sir, this sterilization of nutrient medium is done in autoclave, right? Therefore becomes which sterilization, sir? It is a internal sterilization. Internal sterilization. Whereas sterilization of explant is done with the help of disinfectant solutions. Therefore this is sterilization, sir. External sterilization. Which sterilization? External sterilization. Is it okay? Right? We are discussing steps involved in tissue culture. We completed preparation of nutrient medium, sterilization of nutrient medium, preparation of explant. Now go for number 4, fourth step. What it is? Inoculation. What it is? Inoculation. Sir, what is the meaning of inoculation? Introducing explant onto the nutrient medium is called as a inoculation. Therefore introducing what? Explant onto the nutrient medium. Therefore we are introducing the explant into the nutrient medium. What it is called as inoculation. Sir, this inoculation should be done in aseptic conditions, yes or no? To maintain aseptic conditions during inoculation is done in one chamber by name laminar airflow chamber. Therefore inoculation done inside what, sir? Laminar air flow chamber. Inoculation is done inside what, sir? Laminar airflow chamber, which maintains aseptic conditions. What, sir? Aseptic conditions. Is it okay? Right. This is about what, sir? Inoculation. Now go for the next step. What it is? Incubation. Now go for the fifth step. What it is? Incubation. Now listen here. Sir, now explant is on what, sir? Nutrient medium. Yes or no? On what? Nutrient medium. What is there, sir? Explant. Explant is there, right? Now you leave this explant on nutrient medium. We are leaving the explant on nutrient medium for 3 to 4 weeks of time. Now you tell me, where is the explant on nutrient medium? For how many days? For 3 to 4 weeks of time. In this period of time, explant utilizes the nutrients present in the nutrient medium. Who? Explant. What it is doing? Utilizing. What it is utilizing? Nutrients present in what? Nutrient medium for a duration of 3 to 4 weeks of time. This explant utilizes nutrients present in the nutrient medium, okay, and produces. Explant produces a undifferentiated mass of cells. Produces what, sir? Undifferentiated mass of cells. That undifferentiated mass of cells called as a callus. Therefore what is this callus, sir? Callus is undifferentiated. Callus is undifferentiated mass of cells. Undifferentiated mass of cells called as what, sir? Callus. Now you tell me, sir, what is the meaning of thallus? Sir, thallus means undifferentiated plant body of algae, yes or no? differentiated, undifferentiated, undifferentiated, undifferentiated plant body, undifferentiated plant body of what? Algae, called as what, sir? Thallus. Is this statement okay? Clear? Now listen this statement. Sir, during tissue culture, during what? Tissue culture, where on nutrient medium, what is produced? Undifferentiated mass of cells produced, called as what? Callus. Therefore, callus is a undifferentiated mass of cells produced on nutrient medium during tissue culture. Undifferentiated mass of cells produced on nutrient medium during tissue culture is called as what? Callus. Is this statement okay? Right. Now what we are doing, sir? Once callus is formed, once what is formed? Callus is formed, we are going to add what, sir? Plant growth regulators. Once callus is formed, what are added? Plant growth regulators are added. What are added, sir? Plant growth regulators are added. Plant growth regulators are added. Sir, what are the plant growth regulators we are adding to the nutrient medium after callus formation? After callus formation, we are adding plant growth regulators to the nutrient medium. Yes or no? What are the plant growth regulators we are adding, sir? Auxins and cytokinins. What and what we are adding, sir? Auxins and cytokinins. Auxins and cytokinins. Now listen these statements carefully. Sir, if high amount of auxins, okay, and low amount of cytokinins, high amount of auxins and low amount of cytokinins are added, then what happens, sir? More amount of root is formed. We are discussing about callus, right? Right. Now where is this callus? On the nutrient medium. Callus is on the nutrient medium. Once callus is formed to the nutrient medium, if high amount of auxins and low amount of cytokinins are added, then callus produces what? Callus produces more number of roots. Callus produces what? More number of roots. This process is called as a rhizogenesis. Name itself indicates rhizo means roots. Yes or no? Therefore, what is this process called as? Rhizogenesis. When rhizogenesis takes place, if more amount of auxins and low amount of cytokinins are added, then rhizogenesis takes place. Sir, if low amount of auxins, and high amount of cytokinins are added to what? Nutrient medium. Then callus, what it produces, sir? Then callus produces more shoot. It means more growth of shoot takes place. Growth of shoot will be more. What will be more? Growth of shoot will be more. When growth of shoot will be more? If low auxins and high cytokinins. Is it okay? Therefore, low auxins and high cytokinins are added to the nutrient medium. Then callus, it produces what, sir? It produces more shoot. It means growth of shoot will be more. What is this called as? This is called as collagenesis. What it is called as a collagenesis. Till now, what and what we got? Rhizogenesis and collagenesis. This rhizogenesis and collagenesis together called as organogenesis. What they are called as? Organogenesis or morphogenesis. Organogenesis or what? Morphogenesis. What, sir? Rhizogenesis. Rhizogenesis, right, plus coalogenesis. Rhizogenesis plus coalogenesis is equals to what, sir? Organogenesis or morphogenesis. Organogenesis or what, sir? Morphogenesis. Is these statements okay? Right. Now listen here. Sir, this callus in some cases produces embryo-like structures. In some cases, this callus produces what, sir? Embryo-like structures. When that embryo-like structures are produced, sir, if we are going to add ammonium nitrate, what we are adding? Ammonium nitrate. If we are adding ammonium nitrate and 2,4-D, sir, what is this 2,4-D, sir? 2,4-D, 2,4-dichlorophenoxyacetic acid, 2,4-dichlorophenoxyacetic acid. Its shortcut is 2,4-D. Is it okay? Right, it is a artificial auxin. It is what, sir? Artificial auxin. Sir, if ammonium nitrate and 2,4-D is added to the nutrient medium, then callus produces what, sir? Callus produces embryo-like structures. Callus produces what like structures, sir? Embryo-like structures. What are that embryo-like structures called as? These embryo-like structures are called as somatic embryos. Or it is called as somatic embryos. Sir, somatic embryos are what like structures? Embryo-like structures. Who, sir? Somatic embryos. What like structures? They are embryo-like structures. Is it okay? Right? Now listen here. Sir, what are formed? Embryo-like structures are formed. What are that embryo-like structures called as? Somatic embryos. Sir, somatic embryos, they are embryo-like structures, right? Sir, the somatic embryos, they are also called as embryoids. Also called as what, sir? Embryoids. They are also called as embryoids. Sir, generally embryo means it should be developed from zygote. Embryo should be developed from what? Zygote. But here zygote formed or not formed? Not formed. Therefore, somatic embryos are non-zygotic. Sir, producing root, rhizogenesis. Producing more amount of shoot, coalogenesis. Producing somatic embryos called as somatic embryogenesis. I am not saying somatic hybridization. It is a somatic what? Embryogenesis. Therefore, right, producing what? Somatic embryos. Producing somatic embryos. Producing what, sir? Somatic embryos called as somatic embryogenesis. Therefore, production of somatic embryos, what is that process called as? Somatic embryogenesis. Somatic embryogenesis. Is it okay? Right. Now listen this statement. Sir, what are these embryos? Are somatic embryos. If these somatic embryos, they are coated with sodium alginate. Somatic embryos coated with, they are coated with what, sir? Sodium alginate. Then synthetic seeds are formed or artificial seeds are formed. Therefore, somatic embryos. When the somatic embryo coated with what sir, coated with sodium alginate. Then what are formed sir? Synthetic seeds. A synthetic seed is formed. What is another name for synthetic seeds sir? Artificial seed. See this statement carefully. Sir, generally a seed contains how many parts? Two parts. Seed coat and embryo. What are the two parts in a seed? Seed coat and embryo. What is the first one? Seed coat. Sir, here sodium alginate. What is the second part of a seed? Embryo. Here which embryos are? Somatic embryo. Therefore, somatic embryo when coated with sodium alginate, what are formed? Synthetic seeds or artificial seeds are formed. Right? These synthetic seeds and artificial seeds, easy for storage, easy for transportation. What are easy for storage and easy for transportation? Synthetic seeds or artificial seeds are easy for storage and easy for transportation. Is this statement clear? Right. Now listen this statement. Sir, first we are taking explant, right? What it is sir? Explant. Sir, explant produces callus. Yes or no? Explant produces what sir? Callus. From that callus we are getting a plantlet. We are getting plantlets. Yes or no? Right? Now listen this statement. Sir, explant directly. Explant what sir? Directly. Directly means here what? Without producing callus. Without producing what? Callus. Without producing callus. Explant directly can produce what sir? Can produce plantlets, can produce somatic embryos. Is this statement okay? Explant first it has to produce callus, then only that callus forms plantlets, or that callus produces somatic embryos. Yes or no? But in some cases explant without producing callus, explant directly produces either plantlets or somatic embryos. Is this statement clear? Is it okay? Right. Therefore these are statements concerned to what sir? Incubation. What it is? Incubation. Right. Therefore we completed what sir? Incubation. Now go for the last step, that is acclimatization. What it is sir? Acclimatization. Sir, acclimatization means nothing but adjusting to the new environmental conditions. Sir, tissue culture is carried out in laboratorical conditions. Yes or no? It means now the plantlet, it is in the laboratorical conditions. Is it okay? Right. Before shifting this plant into the open field, we have to increase the resistance of the plant. It means the plant should be resistant to the open environmental conditions. It means we have to make the plant hard. Sir, what is the meaning of acclimatization? Adjusting to the new environmental conditions. It is called as what sir? Acclimatization. Now listen here. Once plantlets are produced, that plantlets present in what sir? Conical flask or test tube. That plantlets present in conical flask or test tube, they are removed. What are removed sir? Plantlets present in conical flask or test tube, they are removed. And these plantlets, they are washed. Why they are washed sir? Because Plantlets are in the test tube. The test tube is with nutrient medium. It means now to the roots of the plantlets, what is present? Nutrient medium is present, right? Therefore, what we are doing, sir? Washing. What? Roots of plantlet to remove nutrient medium. To remove nutrient medium on roots. Therefore, to remove the nutrient medium on roots, we are washing the roots with water. We are washing the roots with water. Roots with water. Now, what we are doing means we are taking this plantlet. What? Plantlet. And we are placing this plantlet in a plastic pot. What is placed? Plantlet is placed in a plastic pot with soil right. What is that plastic pot having soil right? Sir, what is the meaning of soil right, sir? Soil right means nothing but a coconut shell plus other organic matter or other organic material. Is this statement okay? Therefore, we are taking a plastic pot. In that plastic pot, we are taking soil right. And we are placing the plantlet in that plastic pot. What is the meaning of soil right, sir? Coconut shell plus other organic material or other organic matter. Is it okay? Right. Then what we are doing means we are covering this plantlet. Covering the plantlet with a polythene bag. Therefore, plantlet covered by polythene bag. Plantlet covered by what, sir? Polythene bag. Is this statement okay? Clear, sir? Right. Now listen, sir, plantlet it is placed in plastic pot, sir. And this plantlet is covered with what, sir? Polythene bag. This total is done in which room, sir? Glass house. Done inside a glass house. Sir, this glass house is also called as a greenhouse, sir. What is another name for glass house? Greenhouse. Sir, what happens in glass house or greenhouse, sir? Listen this statement carefully. Sir, now plantlet is in glass house or greenhouse? Yes or no? Right. Placing plant inside glass house. Inside what, sir? Glass house or a greenhouse. Helps in hardening. Helps in what, sir? Hardening. Who is becoming hard? The plantlet is becoming hard. Therefore, placing what, sir? Plantlet inside glass house helps in what, sir? Hardening. Hardening means nothing but increasing adaptability. Increasing what, sir? Adaptability. Increasing adaptability. Is it okay? Clear? Therefore, why we are shifting these plantlets to glass house, sir? To make it hard. It means it has to undergo hardening. Hardening means nothing but what? Increasing adaptability of the plant. Increasing adaptability of the plant. Increasing what? Adaptability of the plant. Is it okay? Right. Now listen this statement. Sir, in glass house, light is reduced and high humidity is maintained. In what? Glass house. In what, sir? Glass house. Therefore, inside a glass house, inside what, sir? Glass house, what? Reduced light, high humidity. high humidity. These two are maintained. Therefore, what are maintained inside the glass house are reduced light and high humidity are maintained inside the glass house. Sir, who is providing this high humidity, sir? Polythene bag. Sir, we cover the plantlet with a polythene bag. Yes or no? That polythene bag provides a high humidity to the plantlet. Provides high humidity to the plantlet. Now you tell me, where is a plantlet? The plantlet is inside the glass house. Yes or no? For 1 to 2 weeks, sir. For how many weeks, sir? 1 to 2 weeks. Is it okay? Once the plantlet it undergoes hardening, then plantlet is shifted to the open field. Once plantlet undergoes hardening, then plantlet is shifted to the open fields. Shifted to what, sir? Open fields. Therefore, is these statements clear? Is it okay? Right, sir, in tissue culture we got plantlets in a conical flask or inside the test tube. We are plucking the plantlets, okay, and washing these plantlets with water to remove the nutrient medium which is on the roots of these plantlets. Once these plantlets becomes free from nutrient medium, right, we are taking these plantlets into a plastic pot with the soil, right. Soil, right, means coconut shell plus other organic matter, right. This total process is done in a glass house or greenhouse. Is it okay? And this plantlet is covered with a polythene bag. Is these statements okay? Right. Sir, why we are placing the plantlet in a glass house or greenhouse, sir? For undergoing hardening. Hardening means increasing what, sir? Adaptability. Increasing what, sir? Adaptability. Right. During this hardening process, reduced light and high humidity are provided. Who provides this high humidity, sir? What? Polythene bag provides this high humidity. Inside this glass house or greenhouse, plantlet will be for how many weeks, sir? 1 to 2 weeks. Once the plantlet undergoes hardening, once the plantlet becomes hard, it is shifted to the open fields. Shifted to what, sir? Open fields. Is this statements okay? These are steps involved in tissue culture. Preparation of nutrient medium, sterilization of nutrient medium, preparation of explant, inoculation, incubation, acclimatization. Is it okay? Right? Go for applications of tissue culture, right? Applications of tissue culture. Applications of tissue culture. Now see, during tissue culture we produced more number of plants in less period of time, right, in less space. That's why we are calling it as micropropagation. Therefore, what is the first application of tissue culture? Micropropagation. Micropropagation. What does this micropropagation mean, sir? Producing what? More number of plants. Producing more number of what, sir? Plants in less time and limited space. Therefore, time is less, space is limited. Producing more number of plants, producing large number of plants, producing more number or large number of plants in less time and less space, called as what, sir? Micropropagation. What is called as micropropagation. Number 2, Soma clones. Now you tell me, sir, clone means phenotypically and genotypically identical. Yes or no? Phenotypically and genotypically identical. We are going to call it as a clone. Sir, what is the meaning of Soma clone? Plants produced during tissue culture are identical to their parents, are identical to the original source. So we are calling them as what, sir? Soma clones. Therefore, plants produced when during tissue culture. plants produced during what, sir? Tissue culture are identical to their parents. Identical to their what, sir? Parents. So we can call this plants produced during tissue culture as what, sir? Soma clones. Is it okay? Right. Now go for number 3. Sir, take economically important plants. What, sir? Economically important plants. Which plants? Economically important plants. Economically important plants like tomato, apple, banana, bamboo. Okay, teak or take, eucalyptus. What it is? Eucalyptus. Eucalyptus. Is it okay? Right. Now listen these statements carefully. Sir, economically important plants like tomato, apple, banana, bamboo, teak, eucalyptus, right, are commercially produced. Commercially produced means large scale, in more amounts. Sir, only these plants, sir? Many are there. Some examples we are taking here, right? Therefore, economically important plants such as tomato, apple, banana, bamboo, teak, eucalyptus, etc. They are commercially produced. They are commercially produced. Next, number 4. Go for the 4th one. Listen here. Sir, we can produce viral free plants from viral infected plants. Therefore, what are produced, sir? Viral free plants are produced from viral infected plants. Sir, a plant is infected with virus, sir. Therefore, viral infected plant. From this viral infected plants, we can produce what, sir? Viral free plants. Sir, listen this statement carefully. Sir, I am saying these plants are free from virus. I am not saying that these plants are resistant to virus. Sir, these plants, they are free from virus. But are they resistant to virus? No, they are not resistant to virus. Is it okay? Sir, how we can produce a viral free plant from a viral infected plant, sir? By meristem culture. By which culture, sir? Meristem culture. By meristem culture. Therefore, by meristem culture, by which culture? Meristem culture, a viral free plant. Can we call it as a healthy plant? Yes. Therefore, which plants are healthy plant? Therefore, viral free plants or healthy plants are produced from viral infected plants by which culture, sir? Meristem culture. Sir, in meristem culture, what we are using as an explant? Either axillary bud or apical bud. Is this statement okay? Right? Sir, axillary bud. Even though plant is infected with virus, even though the plant is infected with virus, in that viral infected plant, axillary buds and apical buds are free from virus, because axillary buds and apical buds contains meristematic cells which goes on dividing continuously. Due to continuous division of meristematic cells present in axillary bud and apical bud, axillary bud and apical bud are free from virus even though the plant is infected with what? Virus. Even though the plant is infected with virus. Is this statement okay? Right. Now we are discussing about which cultures are? Meristem culture. Which culture this is? Meristem culture. Right. Now listen these statements. Sir, in meristem culture, what we are using as an explant? In meristem culture, write axillary bud. Is it okay? What is another name for axillary bud, sir? Lateral bud. Which bud? Lateral bud. Right. And apical bud. What is another name for apical bud, sir? Terminal bud. Terminal bud, right? Therefore, in meristem culture, right, this axillary bud and apical bud are used as what, sir? They are used as explant. They are used as explant. They are used as what, sir? Explant. Is this statement okay? Right? Now listen this statement. Okay. Now listen here. We are discussing meristem culture, right? Now listen, even though plant what infected with a virus, even though plant infected with what, sir? Virus, axillary bud and what, sir? Terminal bud. Which bud and which bud, sir? Axillary bud and terminal bud are free from virus. Why, sir? As they contains what? Meristematic cells. Which divides continuously. See this statement? Sir, who are dividing continuously? Meristematic cells. Where that meristematic cells are? In axillary bud and terminal bud. As meristematic cells which divides continuously are present in axillary bud and terminal bud, axillary bud and terminal bud are not with virus, even though the plant is with virus. Therefore, we are taking a plant which is infected with virus. Even though plant is infected with virus, axillary bud and terminal bud of that plant are free from virus, because that axillary bud and terminal bud contains which cells? Meristematic cells, which divides continuously. Due to continuous division of their meristematic cells, axillary bud and terminal bud are free from virus, even though plant is with what, sir? Virus. Is this statements okay? We are discussing which culture, sir? Meristem culture, right? Which culture it is? Meristem culture. Therefore, scientists succeeded right, in producing what, sir? In producing viral free plants. Scientists succeeded in producing viral free plants in case of banana, sugarcane, potato. Is it okay? Banana, Musa paradisiaca. Sugarcane, Saccharum officinarum or Saccharum barberi. Potato, Solanum tuberosum. Therefore, see this statement carefully. Sir, scientists, they were succeeded in producing viral free plants in the case of banana, sugarcane, potato, etc. By which culture? Meristem culture. By which culture? Meristem culture. What is the explant? Axillary bud or terminal bud. What are produced? Viral free plants are produced. In case of which plants? Banana, sugarcane, sugarcane, potato, and etc. Is this statements okay? Right. These are applications of what, sir? Tissue culture. These are applications of tissue culture here. Note. Number 1. 3 lysine-rich maize varieties. Which varieties, sir? Maize varieties. Rich in what, sir? Lysine. How many varieties we are discussing? 3 varieties. 3 lysine-rich maize varieties are developed in India. They are developed in what, sir? India. What they are? Protina, Shakti, Rathan. Protina, Shakti, Rathan. These 3 are lysine-rich maize varieties that are developed in India. Sir, lysine, it is a protein or amino acid? It is a amino acid. Which amino acid? Basic amino acid. Lysine is a basic amino acid, right? Next, number 2. Mushroom is a source of protein. I am not saying that it is the only source of protein. Mushroom is also a source of what, sir? Protein. A source of what, sir? Protein. Sir, mushroom, right? What it is? Mushroom, right? Sir, this mushroom comes under, right, class what, sir? Mushroom comes under class Basidiomycetes. This we read in first year. Sir, mushroom is algae or fungi, sir? It's a fungi, right? Mushroom is a fungi. Mushroom is a fungi, right? Sir, edible mushrooms are there, poisonous mushrooms are there, right? Edible mushrooms are there as well as poisonous mushrooms are also there. I am speaking about edible mushrooms. Edible mushrooms comes under genus what, sir? Edible mushrooms comes under genus Agaricus. Comes under genus what, sir? Agaricus. Comes under genus Agaricus. What? Edible mushrooms comes under genus Agaricus. Is this statements okay? Right. Next one. Sir, we discussed 3 types of selections: pure line selection, mass selection, clonal selection. Yes or no? Right? Pure line selection. What? Mass selection. Pure line selection, mass selection. Next one is what, sir? Clonal selection. Clonal selection. Sir, by pure line selection, a variety is developed by name TMV3. It is a variety of groundnut. It is a variety of what, sir? Groundnut. Arachis hypogaea. Therefore, by pure line selection, a groundnut variety is developed by name TMV3. Okay. Now go for mass selection. By mass selection, a variety of bajra is developed by name Pusa Moti. What it is? Pusa Moti. It is a variety of bajra. It's a variety of bajra. Now go for clonal selection. Sir, by clonal selection, by what, sir? Clonal selection, a variety of potato is developed by name Kufri Red. Kufri Safed. SAFED. Sir, this Kufri Red and Kufri Safed, these 2 are varieties of what, sir? Potato. These 2 are varieties of what, sir? Potato. Is this statements okay? Clear? Right. Sir, we are discussing some important statements in note. We completed 1, 2, 3. Right now we are going for number 4. Anther culture. Go for what, sir? Anther culture. Anther culture. Sir, anther culture is also called as pollen grain culture. Also called pollen grain culture. Sir, it is also called as a androgenic haploid culture. Androgenic haploid culture. Is it okay? Therefore, anther culture is also called as a pollen grain culture. It is also called as androgenic haploid culture. Sir, in anther culture, what we are doing, sir? Take a anther. Sir, we are taking what, sir? Anther of a plant species. From plant species, we are taking anther. If you culture this anther on nutrient medium, we are culturing what? Anther on what, sir? Nutrient medium. Then this anther, when cultured on nutrient medium, this anther produces haploid plants. What are produced, sir? Haploid plants are produced. What are produced? Haploid plants are produced. This is called as which culture, sir? Anther culture. Therefore, culturing anther of a plant species on a nutrient medium to produce haploid plants called as which culture, sir? Anther culture. This is called as which culture? Anther culture. Is this statement okay? Right? Now, listen this statement. Sir, which plants we got here? Haploid plants. Haploid plants. Haploid plants produced by anther culture. Haploid plants produced by anther culture are called androgenic haploid plants. What they call as androgenic haploid plants. Sir, these androgenic haploid plants have some variations. Androgenic haploid plants have some variations. Sir, what are these variations called as? These variations are called as gametoclonal variations. What are these variations called as? Gametoclonal variations. Is this statement okay? Therefore, sir, androgenic haploid plants means the haploid plants produced by anther culture. These androgenic haploid plants, they have some variations. What are that variations called as? Gametoclonal variations. Gametoclonal variations. Now listen this statement. Sir, when we are culturing anther of a plant species on nutrient medium, right, this anther produces which plants, sir? Haploid plants. What it produces, sir? Haploid plants and some diploid plants. And some what, sir? Diploid plants. Therefore, in anther culture, among haploid plants, you can see some diploid plants. Is this statement okay? Right? Therefore, in which culture? Anther culture. In anther culture, among which plants? Among haploid plants, some diploid plants are seen. Sir, among haploid plants, which plants are seen, sir? Some diploid plants are seen. Sir, what is the source of this diploid plant, sir? What, sir? Source of diploid plants. In which culture? Anther culture. Is a anther wall. In first year, we read anther wall, layers of anther wall, epidermis, endothecium, middle layers, tapetum or tapetum, right? Therefore, sir, what is the source of diploid plants in anther culture, sir? Anther wall. Sir, generally the layers of anther wall, are they haploid or diploid, sir? They are diploid. Is this statement okay? Right? Now, listen this statement. In India, right, anther culture. What? Anther culture first done by Guha and Panchanan Maheshwari to produce what, sir? Haploid plants of Datura inoxia. What it is? Datura inoxia. See this statement. In India, this anther culture was first done by who, sir? Guha and Panchanan Maheshwari to produce which plants? Haploid plants. Haploid plants of which plant? Datura inoxia. Sir, this Datura inoxia belongs to which family? Solanaceae family. Datura inoxia belongs to which family? Solanaceae family. Is this statement clear? Therefore, by anther culture, haploid plants are produced. Okay. Among that haploid plants, here and there you can see the diploid plants also. But haploid plants are produced. Is it okay? Right. Haploid plants are produced even by culturing unfertilized ovules. Sir, here I am taking which ovules are unfertilized ovules. Unfertilized ovules. Listen this statement. Sir, ovule is a diploid, right? Ovule is a diploid. But is this ovule fertilized or unfertilized? Unfertilized. Sir, a unfertilized ovule contains right, haploid cells. Unfertilized ovule, right, it contains embryo sac. That embryo sac contains egg cells, synergid, antipodals, yes or no? Therefore, it contains which cells are haploid cells. It contains which cells are haploid cells. From these haploid cells, from which cells, sir? Haploid cells, what are produced? Haploid plants are produced. Therefore, unfertilized ovules contains haploid cells. Right, from this what? Haploid what, sir? Cells. From which cells, sir? From these haploid cells, what are produced? Haploid plants produced. Is this statement okay? Now listen here. Haploid plants produced from haploid cells of unfertilized ovules called as gynogenic haploids. Therefore, haploid plants produced from what, sir? Haploid cells of unfertilized ovules called gynogenic haploids. Therefore, gynogenic haploids are which plants? Haploid plants. Produced from where? Haploid cells of unfertilized ovules. Is this statement clear? Okay, right? Embryo culture. What it is? Embryo culture. Embryo culture. Sir, take developing seeds. What we are taking? Developing seeds. From this developing seeds, young embryo is isolated. Sir, what is isolated, sir? Young embryo is isolated. Young embryo is isolated. Sir, this isolated young embryo, when grown on a nutrient medium, when grown on what, sir? Nutrient medium, it produces what, sir? It produces young seedling. Produces what? Young seedling. Therefore, what is produced? Young seedling is produced when young embryo of a developing seed is grown on a nutrient medium. This culture is called as embryo culture. Is this statement okay? Right. Therefore, what is obtained? Young seedling is obtained when young embryo is grown on a nutrient medium. What is grown? Young embryo. This young embryo belongs to what? A developing seed. Therefore, from a developing seed, young embryo is isolated. And this isolated young embryo, when grown on a nutrient medium, it produces a young seedling. This is called as which culture, sir? Embryo culture. It is called as embryo culture. Now, what are the uses of this embryo culture, sir? Listen. In some interspecific crosses, in what, sir? In some interspecific crosses, endosperm of what? Endosperm of a hybrid developing seed degenerates early. So, young hybrid embryo does not gets food. Does not gets what, sir? Food. Listen these statements carefully. Sir, we are discussing about interspecific crosses. Which crosses? Interspecific crosses. Sir, interspecific crosses, a type of hybridization. Yes or no? Type of hybridization. So I am calling these seeds as a hybrid developing seed. Therefore, hybrid developing seeds produced by interspecific crosses. These hybrid developing seeds, what they contain, sir? They contains a young hybrid embryo. Contains what, sir? Young hybrid embryo. Who is having young hybrid embryo? Hybrid developing seeds contains young hybrid embryo. Sir, who provides food to the embryo, sir? To the embryo, endosperm provides food. To this embryo, who is providing the food? Endosperm. But this endosperm has disintegrated in early stages. If endosperm disintegrates in early stages, then embryo, does it get the food? It won't get the food. Therefore, embryo starts disintegrating. Yes or no? In such cases, embryo is isolated. Young hybrid embryo is isolated from hybrid developing seed. And that young hybrid embryo is grown on a nutrient medium to produce what's our young seedling. Is this statement okay? It means here embryo is rescued. Embryo rescue. It means right, we are applying what? Embryo culture. Application of embryo culture to prevent abortion of young embryos is called as embryo rescue. Is this statement clear? See these statements carefully. Sir, in interspecific crosses, okay, a hybrid seed is produced. A hybrid seed is produced. That hybrid seed is having young hybrid embryo. this young hybrid embryo gets food from endosperm, but this endosperm has disintegrated in earlier stages. So this young hybrid embryo is not getting food from the endosperm. So this young hybrid embryo starts disintegrating. So in such interspecific crosses where endosperm disintegrates in earlier stages, in such cases the young hybrid embryo is isolated, right, and grown on a nutrient medium. Is these statements okay? What is this culture called as? Embryo culture. Therefore, application of application of embryo culture. Application of embryo culture to prevent what? Abortion. To prevent abortion of a young embryo is called embryo rescue. Is this statement okay? Clear, right? Number 2, go for the second application. For the second use of this embryo culture, listen. Sir, in case of orchids, right, or orchids, in case of orchids, right, no stored food in the seed. Therefore, in seeds of orchids, no stored food. So embryo, does it get the food? No. So embryo does not gets food. Embryo is not getting the food. In such cases, what we are doing? We are isolating the embryo from the orchid seeds and we are culturing this embryo on the nutrient medium. Which culture it becomes? Embryo culture. Which culture it becomes? Embryo culture. Number 3. Sir, in some plant species, seeds are dormant. Seeds are dormant. It means seed is unable to germinate. Seeds are dormant. Seeds are unable to germinate. Okay. Therefore, in some plant species, seeds are dormant due to, why sir? Due to inhibitors. It means these inhibitors present in the seed, they are not allowing the seed to germinate. In such cases, you isolate the embryo from that dormant seed and culture it on the nutrient medium. Becomes what, sir? Embryo culture. Sir, seed is unable to germinate. It means seed is undergoing dormancy due to the presence of what? Inhibitors. It means inhibitors are not allowing the seed to germinate. In such cases, the young embryo is isolated from that dormant seed, and that young embryo, when cultured on the nutrient medium, right, it produces a young seedling, right? We can call it as which culture, sir? Embryo culture. Therefore, is these statements okay? Right? This is concerned to embryo culture, right? Thank you.